Hello, everyone. Thank you so much for joining us today. My name is Mina Nabavi, and I'm a program manager with the Education Core with the UAB CounterACT Research Center of Excellence in Arsenicals. Welcome to today's Grand Round seminar on arsenic and NRF2 signaling in cancer and diabetes. For those of you not familiar with our center, it is led by Dr. Mohammed Athar, and the goal of the center is to develop mechanism-based, highly effective medical countermeasures against arsenical chemical warfare agents. For more information on the center or its activities, please visit our website at uab.edu/arsenicals or send us an email at as33@uab.edu. On our website, you will find links to recordings of past Grand Round presentations and our podcast, as well as information on future Grand Rounds as they are announced. And now I'd like to introduce our speaker today, Dr. Donna Zhang. Dr. Zhang received her PhD from New York University in 1997. After a brief postdoctoral fellowship at the DuPont Haskell Laboratory, she joined the University of Arizona as an assistant professor in 2005, where she is currently a professor in the Department of Pharmacology and Toxicology in the College of Pharmacy. She is also the Musil Family Endowed Chair in Drug Discovery and the Associate Director of the Superfund Research Program at the University of Arizona. Dr. Zhang's research program focuses on investigating the key physiological and pathological roles of the NRF2 KEAP1 ARE signaling pathway and mediating disease. She has published over 130 well-received papers and has made seminal contributions to both the mechanistic understanding of NRF2 regulation and the role of NRF2 in human diseases. Welcome, Dr. Zhang. We're glad you could join us today.
Thank you so much, and especially thanks Mina and also Director Arta for inviting me. And Mina made this so wonderful. Really appreciate it. It is my honor to give a seminar to your center. So my lab has been working on a protein called NRF2 from mechanistic study how NRF2 is regulated to its role of NRF2 in human diseases. And also, we are very interested in environmental toxicants, arsenic. So I guess this is the common ground of our center and this center. So, so first of all, let me give some historical overview of the pathway. So yeah, we are very fortunate in the NRF2 field, and show you here that every year the publication under explosion, and the NRF2 is getting more and more popular. So back almost 2 decades ago, this is a field started. So mainly by scientists in chemoprevention field. So one of the pioneer I must mention is Paul Terry. So his lifetime was dedicated to understanding how to use cyclic compound to reduce incidence of cancer. So here he was looking at the broccoli sprouts, and inside this broccoli sprouts, there's a compound called sulforaphane. Here is the structure. So consumption of sulforaphane can prevent you from cancer. So since then, many cyclic compounds. So here is the structure of all those ingredients from those healthy food. Consumption of those food can protect you from cancer. So those chemopreventive compounds have one common property, that is they all interact with the sulfur hydrosulfur group of the proteins. So then in about 1990, and scientists try to figure out how this cyclic compounds can induce a set of gene expression. So those genes are detoxifying enzymes and antioxidant. So those are the classical name, and later on you will know they are classical NRF2 target genes. So then in 1997, Bill Fall at Wisconsin analyzed the common sequence in the promoter of those genes and identified a consensus sequence such as ATGA followed by 3 and then GC. So he named antioxidant response element, so ARE. So then the next question is looking for what are the transcription factors that binds to ARE. So that was around the year 2000. And so there's 2 NRF2 knockout mice was made. One is made by San Francisco, UCSD, Dr. Kong, and another is by Musil Yamamoto. So Tom was doing chemoprevention study in China. On his way back, he got Musil Yamamoto's NRF2 knockout mice. So use this NRF2 knockout mice, Tom's group clearly defined that NRF2 is the transcription factor that binds to ARE. So as this landmark paper stated that NRF2 knockout mice has decreased basal expression of phase 2 detoxifying enzymes. Therefore, those mice are super sensitive to chemical carcinogen. And if you feed those mice with NRF2 inducers such as sulforaphane, the induction by those chemopreventive compounds is blocked. So in NRF2 knockout mice, those chemopreventive compounds lost activity. Another significant events in the field was in 1999, Ken Ito in Yamamoto's lab identified a negative regulator of NRF2 called KEAP1. So in that gene and development paper, they present a very simple model to explain how KEAP negatively regulates NRF2. So they think it's physically retain NRF2 in the cytosol, so block NRF2 transcriptional activity. So KEAP contains a couch domain, which is the domain bind to the N terminal of NRF2. So this is the couch domain and also the BTP and the intervening region. So that was 1999. Basically, this was the starting of the NRF2 field. So I started interested in this pathway in year 2000. So so now I'm going to talk about the work from my lab. So part 1, I will simply will briefly talk about how NRF2 is regulated. Again, so far, KEAP1 is the primary negative regulator of NRF2. Actually, it's the E3 ubiquitin ligase causing NRF2 degradation. So there's a canonical way and a non-canonical way, which is very important in distinguish good and bad aspect of NRF2. So, and in part 2, I will talk about the role of NRF2 in human disease, and so how canonical activation of NRF2 is good for cancer prevention, as originally the field started. And later on, I will talk about how arsenic induced NRF2 in a better way, the dark side of NRF2, and it's more mimicking the KEAP mutation in tumor. So first, back to year 2000, when I started interested in this pathway, so I analyzed the data sequence of KEAP1 and come out this hypothesis that KEAP probably is an E3 ubiquitin ligase. That is a curling-based E3 ubiquitin ligase. So all the data supporting this model KEAP actually is a substrate adapter protein that brings in a choline 3-based E3 ubiquitin ligase called NRF2 degradation. So this pathway, this way is called canonical activation of NRF2. So in normal case in your body, NRF2 is expressed ubiquitously in any organs and cell types. However, the expression level is super low. That's why you need to boost NRF2 periodically for chemoprevention. So normal case, KEAP constantly targets this NRF2 for ubiquitination and degradation. So after you eat broccoli sprouts, if enough sulforaphane there, then there's a conformational change in this E3 ubiquitin ligase. So NRF2 is no longer ubiquitinated. Now the newly synthesized NRF2 is stabilized because couldn't find KEAP1 for degradation, right? So excess amount of NRF2 moves into nucleus where it finds the ARE enhancer sequence, form heterodimer with small map protein to direct transcription of NRF2 target genes such as NQ1, GCLM, HO1, and AKR1C1. Those are classical NRF2 target genes. So this mechanism called canonical NRF2 activation, which is good for chemoprevention. So next, I'm going to talk about a non-canonical mechanism of activation. This actually is how arsenic, environmental toxicants, induce NRF2, resulting in prolonged NRF2 activation, which is bad because NRF2 is a critical transcription factor. So on and off have to be regulated tightly. So arsenic resulting in prolonged NRF2 activation, which is bad. So this is the mechanism we call the non-canonical NRF2 activation by arsenic. This is still KEAP dependent. So initially, because the Yamamoto's structure finds this 2 KEAP1 shown in green here bind to 1 molecule of NRF2, and there's 2 binding sites like hinge and the latch. So one is strong binding, ETG is a very strong binding domain, and another is a weak binding, DLG. So weak side is very easy to be opened up. So we find that P62 have a similar domain, actually STGE, similar to ETGE. So P62 itself able to bind to couch domain of KEAP1. So disrupt this E3 ubiquitin ligase complex, resulting in NRF2 activation. So here is the data. We use wild type P62 and mutated P62 show that only overexpression of wild type P62 induce NRF2 and its target gene. So what is P62? P62 is a very important cargo substrate for autophagy, for autophagosome. So autophagosome is formed first as a phage formation. After phage form, it's elongated to form a double membrane autophagosome. So within this double membrane autophagosome, there's 2 important proteins I need to mention. One is show you green here is P62. It brings in the ubiquitinated cargo or like damaged organelles or protein aggregates into this autophagosome. The next step is autophagosome merge with lysosome to form autolysosome. Then the inside those organelles or protein aggregates can be degraded. So this is called autophagic pathway. So what we find is, so if you overexpress P62, So P6A2 is inside this autophagy zone. This will disable KEAP1, will sequester KEAP1 into this wrong compartment. So KEAP can no longer functioning as easily recruiting ligase to target NRF2 for degradation. Okay, so that is a non-canonical mechanism. It is how arsenic activates NRF2, as I show you in the next slide. So that is the arsenic treatment. So arsenic block here. So blocks this fusion of autolysosome with autophagosome with lysosome. So blocks this step. So how we find this? This is the evidence. We use this LC3. So LC3 is a protein attached to the autophagosome membrane. So use this IFP GFP. GFP is acidic labile. So if this merged, then GFP will be gone. So will be red. So red puncta indicating autolysosome. And the yellow means both IFP GFP is expressed, indicating autophagosome. So normal case, if you overexpress this LC3, so it's just in the side, in the whole cell cytosol. Under starvation is the inducer of this autophagic pathway, will lead to autolysosome formation. So you will see red and yellow. You see both autophagosome and autolysosome. Sulforaphane, which is the canonical NRF2 inducer, didn't do anything for this autophagic pathway. However, if you treat with arsenic, 500 nanomole, so we are working with very low dose of arsenic. So for environmental relevant, arsenic is everywhere, right, on the earth, in the water. So we use very low dose, which is different than probably your center is doing. Your center is doing chemical warfare agent. So probably the dose is high, but we are working on environmental arsenic exposure. So with very low, as low as 100 nanomole of arsenic, completely blocks the fusion, basically blocks this step, because you see perfect merge. So you only see autophagosome, not autolysosome. So you only see this autophagosome, not autolysosome. Okay. So basically this is the model. So P62 after arsenic treatment blocks the fusion. So P62 sequester KEAP into this autophagosome, resulting in prolonged activation of NRF2. And this is the data actually we show, we make CRISPR knockout of KEAP1, put back in the cysteine 151. So canonical chemoprevention compounds such as sulforaphane induce NRF2 in a KEAP cysteine 151 dependent manner. However, arsenic induction does not require cysteine 151 because it's the P62 dependent, sequester entire KEAP1 into autophagosome. So that is the summary for the mechanistic part. In normal case, the intermittent activation of NRF2 is good for cancer prevention. However, in certain cancer type, because of somatic mutation in KEAP1 or in NRF2, resulting in disruption of the KEAP NRF2 binding, so KEAP can no longer regulate the NRF2 protein level. So NRF2 is accumulated, so resulting in constitutive activation of NRF2. So NRF2 is always higher there. Then in the case of arsenic, because of blockage of the fusion of autophagosome with lysosome, so KEAP1 is accumulating, and P62 sequester KEAP1 into autophagy zone, resulting in NRF2 activation. So that is the molecular mechanism of how NRF2 is regulated, and mainly in a KEAP-dependent manner. Canonical NRF2 activator induced NRF2 requires all the cysteine residues in KEAP1. However, in arsenic case, there's no cysteine 151 needed. It requires the P62 autophagy pathway. So over the years, especially the first decade, everybody think NRF2 is great. Induced NRF2 can prevent us from any reactive oxygen species related disease, right? ROS, reduce ROS for disease prevention. So people are more focused on the NRF2 inducer. So to identify NRF2 inducer is easy because any electrophile compounds can induce NRF2, right? They are reactive with cysteine residues of KEAP1. So one inducer called dimethylfumarase has been in the market, has been FDA approved NRF2 inducer. However, it's a very weak NRF2 inducer. My lab also tested in our group. It's very weak NRF2 inducer, and so there's also a couple of publications indicate even in NRF2 knockout mice, they still have some efficacy. So it means it's NRF2 independent mechanism. So if you want to know more, there's a lot of dietary compound or phytochemicals. They are NRF2 inducer, and people are doing it for clinical trial. So to me, the most critical thing is to find a NRF2 specific inducer for disease prevention. So we, my lab have been using NRF2 inducer for different disease models, such as for colon cancer prevention, and also diabetes and diabetic complication, and diabetic wound healing. So we did show that induced NRF2 also help diabetic wound healing. So let me now switch to the dark side of NRF2. So the second decade of the field now, especially now, because so many high-profile cancer research, such as Tyler Jack, also interested in this pathway. So now, from NRF2, because of both bright side and the dark side, so now they actually start to call NRF2 oncogene, which is really, I don't know whether I should call NRF2 oncogene or not, but once cancer cell is already transformed, high level of NRF2 is protecting cancer for surviving and promoting tumor growth. So we presenting this dark side of NRF2 back in 2008. So at that time, it is very clear to me that NRF2 is a cell surviving signal, right? It's protect cell. Whatever it's normal cell or it's cancer cell, induction of NRF2 is come for protection, okay, against the cell death. So we presenting this NRF2 enhanced resistance of cancer cells to chemotherapeutic drug, the dark side of NRF2 in year 2000. But now it's getting more and more popular. There's many publications, and my former graduate student, Musil, have this nice article in Cancer Cell presenting that in normal cell, induction of NRF2 is good for prevent cell transformation. However, once cell is already transformed, KEAP-induced NRF2 will protect the cell surviving under and the detrimental environment of the cancer, and also promoting cell proliferating. And there's also lots of paper indicate that NRF2 induction help cancer cells spread metastasis to other organs. So from here is more like dark side of NRF2. So when should induce, how long should induce it's of NRF2 in cancer case is very complicated issue. So then in 2014, this paper, they analyzed the KEAP1 mutation or NRF2 mutation and find in lung squamous. So that is the KEAP1 mutation and NRF2 mutation. And they usually mutually exclusive because you don't need to mutate both NRF2 or KEAP1 to get constitutive activation of NRF2. So in lung adenocarcinoma, almost 1/3 patient has KEAP1 mutation. Okay? So on the head and the neck and bladder cancer. So if you see the enrichment data and the KEAP mutation is in cancer is as popular as those famous P53, KRAS, PTEN. So that's why now NRF2 is also being considered as an oncogene. So constitutive activation in cancer cells is really detrimental. So since the year 2000, when I got an assistant professorship in Missouri, University of Arizona, I was already very interested in looking for NRF2 inhibitor, while the other labs still looking for inducer for cancer prevention. So we found the very first NRF2 inhibitor, pathway inhibitor. It's not directly targeting NRF2, it's inhibiting NRF2. So this is a Brusatol. So at 40 nanomole, it's induced LC50 is half. Okay. So use this, and we use, of course, cell line, and also now A549 xenograft. And without treatment, this is without treatment. Tumor is already grown to 800. So with single either Brusatol treatment or cisplatin, this is chemo drug, right? Single treatment is in the middle, both treatment. So our notion is to inhibit NRF2 during chemotherapy is important. So use NRF2 inhibitor as adjuvant. Okay. So here is the data. Then because of this work, I got NCI grant. So this is the AIM-3, use a better lung model. This is Tyler Jack's KRAS G12D model, and with CLE to activate G12D. So once KRAS is activated, it get lung tumor. So if you look at this, without any treatment, mouse all in this group dead at about 11 to 12 months. Single treatment of cisplatin or Brusatol, about 20% survive the lifetime. Co-treatment, 40% of mouse in the group survived the lifetime. So basically indicating that the importance of NRF2 inhibitor. So that is the summary for NRF2 and the cancer. So it's very complicated case. How long you need to induce the NRF2 and when you need to inhibit the NRF2. So it's all in this review article because it's NRF2 participating in every single hallmark of cancer. So now let me talk about arsenic, which is more relevant. So as I said, we're interested in very low dose of environmental arsenic exposure, and to this group of people, so you all know what is arsenic, and arsenic in particles. So we drinking water and food. So we ingest or inhale arsenic all the time. And the US map, even some the EPA limit is 10 ppb, but lots of places like show in red in the map have higher than the EPA recommended amount. And also in India, there's arsenic amount is very high in well water. So that's the correlation. So arsenic, today I'm only going to talk about arsenic and the type 2 diabetes. That's a very new field. And there's a human epidemiology study indicating that there's arsenic contamination, arsenic link with type 2 diabetes. So we did a study, use different group of mouse. So wild type, NRF2, try to see whether it's NRF2 dependent, right? Because we think the arsenic cause prolonged NRF2 activation is associated with diabetes. And also non-canonical activation requires P6Day2. So we have P6Day2 and a double knockout. And we give arsenic through drinking water, 25 ppm, okay, for 10 weeks. So we did metabolomic and ionasic. Also first the phenotype. So after arsenic in drinking water, you see the wild type do see significant impairment of glucose tolerance, and that is NRF2 and P6Day2 dependent. And also arsenic exposure causes insulin resistant. So here's the wild type, insulin resistant. So we did a metabolomic and ionasic, and then my colleague, bioinformatics guy, and analyze. It's very complicated story. So he analyzed, and then one thing caught our eye, that arsenic induce this 4 gene expression. So this is the control, each is individual mouse. Arsenic treated mouse, 1, 2, 3, 4, 5. So all induce this gene expression. However, it's NRF2 dependent. So it's disappear in NRF2 knockout and also disappear in P6Day2 knockout. So we're super interested in this arsenic induction of those 4 genes. So it turns out, yes, all those 4 genes contains AIE. So this is the AIE search. So to me, unless you show mutation of identify AIE to define, yes, this is NRF2 targeted gene. So define AIE and mutated AIE no longer work. So this all show that arsenic, 2 doses of arsenic, dose dependently induce these 4 genes. Right. However, so this we use liver slides, so ex vivo liver slides in cell culture dish media and put arsenic. So NRF2 knockout, so liver slides from NRF2 knockout completely gone. So indicating yes, NRF2 dependent. And also we use TRIP-seq and identify their true AIE in these 4 genes. So if you look at the function of this gene, you will see that they all related to sugar metabolism. So those are the 3 genes in the sugar glucose. So polio pathway. This is the polio pathway, and also the HNF4A, which is controlling a transcription factor itself, controlling gluconeogenesis. Again, it's to determine the glucose amount. So that we used labeled sorbitol, okay, to see how sorbitol is metabolized into glucose with without arsenic treatment. So here is without arsenic treatment. Here is arsenic treatment. So you see that more glucose is produced, more labeled glucose produced from labeled sorbitol. So indicating basically your liver probably after arsenic treatment, after ingestion or inhale arsenic, your liver produce more glucose into bloodstream. So it's the diabetic phenotype. So the conclusion of this study is inorganic arsenic increased the carbohydrate flux through NRF2 mediated upregulation of the polio pathway and gluconeogenesis pathway. So the conclusion for this arsenic diabetes is inorganic arsenic mediate non-canonical activation of NRF2 because it's the P62 dependent. And the glucose intolerance and insulin resistance induced by chronic arsenic exposure is mediated by genetic aberration of NRF2 and the P62. Inorganic arsenic alters expression of clinical genes controlling glucose fructose metabolism. So that is a novel molecular basis of how arsenic promoting diabetes through prolonged activation of NRF2 through the oxide of NRF2. So finally, I would acknowledge those people. So my collaborator outside and inside university and my group. Lastly, I would like to acknowledge during my career, so I majority funding got from NIEHS, and currently I have R35 from NIEHS, and also for diabetes work, I got from NIDDK. For cancer work, I got funding from NCI.
So I'd like to open it up now. So if you have a question, please feel free to unmute and ask Dr. Zhang directly.
Dr. Zhang?
Hey,
Hi, this is Veena Anthony. Hi. Superfund at UAB. So I just wanted to touch base and ask you this question. Do you think it increases, arsenic increases insulin resistance and thus cause diabetes? Or has it been found in metabolic syndrome? Because on the clinical side, we don't necessarily associate arsenic poisoning. We don't even test for arsenic in most of our patients who are diabetic.
I see. Yeah, so I don't think there's a lot of patient data from US. So those epidemiology study is usually done with the like Bangladesh, Indian, right? They only find a correlation, like yeah, correlation study. So, but nobody really done anything about mechanistic how arsenic promoting diabetes. So it's yeah, it's quite difficult to study, right? Except a correlation study.
Imagine, yes.
Because the human exposed to not just the arsenic, everything, right? It's that's the problem, yeah. So in lab, you know, if mouse, not only in so mouse in cancer, arsenic study in mouse is so difficult because mouse metabolize arsenic differently.
Yes.
So they think probably because mouse have less methyl transferase. So inorganic arsenic gets into our body we can metabolize into MMA and DMA
Right.
methylated form.
Yes.
But a mouse is quite difficult. So that's why mouse requires higher dose.
Mhm.
So it's always the big challenge in arsenic study. In
Mhm.
actually any metal, because it's, human exposure is not just a pure one metal, right?
Of course.
Yeah.
Combined
So
exposures, yeah.
yeah,
Thank you so much, Dr. Zhang.
thank you.
All right. Well, Dr. Zhang, I want to thank you again for presenting for our group. And I see several thank yous coming into the chat. And thank you all for attending. We look forward to seeing you at our next Grand Rounds. Please stay tuned to our website. We are recording several podcasts over the next few weeks, so those will be available on our website. And additional Grand Rounds will be posted there as well. Thank you so much and have a great day.